In our previous video, we discussed types of cell. So this is a continuation of the previous video. We will be discussing functions of major organelles in animal cell. An animal cell is one example of eukaryotic cell. So let's look. Organelles, basically, they are cellular components that perform specific job for the cell. Every other thing you see here, they are all organelles. These are the names of major organelles that you find in eukaryotic cells, specifically animal cell. So let's look at the first organelle. This structure is called the cell membrane. Cell membrane. This entire structure around here is called the cell membrane. In some books you call it plasma membrane. So don't be surprised if you see that on your test. So the function of the cell membrane is to regulate the entrance and exit of material in the cell. In order for things to enter the cell and leave the cell, it is the cell membrane that regulates that. We also call it the gatekeeper because it's regulating every other thing that enters and leaves the cell. When you leave the cell membrane, you're going to enter directly into the cytoplasm. The cytoplasm is made up of a gel-like fluid that fills the entire cell and hold the cellular contents. Most of the organelles you find in the cell, they are suspended in the cytoplasm because the entire cytoplasm has a fluid that is made up of a gel-like substance. The next organelle, once you get into the cytoplasm, in fact, if you want to swim, you can even swim in the cytoplasm because there is a fluid there, a gel-like fluid. There is a control center, which is the nucleus. So if you are fortunate to enter the cell, right after swimming in the cytoplasm, you're going to meet the boss, which is the nucleus. We call this nucleus the control center of the cell because it is the organelle that houses the genetic material. Don't be surprised if you hear the genetic headquarters of the cell. That would be the nucleus, because the nucleus holds the genetic material of the cell, so it controls everything. Right after the nucleus, there is a tiny little structure in the nucleus that we call the nucleolus. The function of the nucleolus is to produce ribosome. We sometimes call it ribosomal factory. So there are so many factories in the cell. That's one of the factories. So in the nucleolus, there is a factory they call the ribosomal factory. Right after the nucleus, when you are coming out, you will see ER, endoplasmic reticulum. There are two types of ER. We have a rough ER and a smooth ER. There's a difference between the rough ER and the smooth ER. The function of the rough ER is that it has ribosome attached to it and is used for protein synthesis to make protein. But the smooth ER lacks ribosome. The tiny little structure you see here are the ribosome that are attached to this ER. Because ribosome is a protein and it is rough, whenever it's attached to an ER, we call it rough ER. But when there's no ribosome attached to any other endoplasmic reticulum, that is a smooth ER. The function of the smooth ER is to synthesize lipid. For example, most of the lipid that you use here to repair the cell membrane of the cell when they are damaged, they are synthesized into this organelle called the smooth ER. After the smooth ER, we see that there is a guy here called ribosome, the tiny little structure called ribosomes. The ribosome is also another factory in the cell. We call it protein factory because the ribosomes are the ones that are used to synthesize the protein in the cell. For example, if you cut your hair needs to grow back. In that process, the ribosomes are the ones that are going to synthesize the new protein, which is your hair, so that you can be able to grow back. And when the ribosome synthesizes the protein, it has to modify it it will send it directly to the Golgi apparatus. So the protein that is leaving from the ER into the Golgi apparatus, the function of the Golgi apparatus or the Golgi body is to modify the protein. It carries modification by adding the glucose, by adding protein and other things. This factory, which is the Golgi apparatus, is just to modify the protein that has been produced by the ribosome in the rough ER. Then right after this guy modify the protein, you can modify it by packaging, sorting, and adding additional material like glucose and protein to that particular protein so that it can be sent to that very destination. When the Golgi apparatus modify the protein, it gives it directly to the vesicle, and the vesicle will transport it directly to this membrane. This vesicle will fuse with the membrane to send the protein out through exocytosis. And if the protein is supposed to be used by the cell, then it will be used by this specific cell. But if it's supposed to leave the cell to go wherever it's supposed to go to perform its function, they will be sent there. For example, if you produce an enzyme that have to be sent to the intestine to catabolize those nutrients that you take in, they will be sent directly to the intestine. And who will take it from the Golgi body and then fuse it with the cell membrane? That will be the vesicle. That's what we have in here. Lastly, we have the mitochondria. The mitochondria is known as the powerhouse of the cell because it is the mitochondria that produce our energy. Majority of the energy that is produced in animal cell, specifically, it is made in the mitochondria. Because it is in the mitochondria that the product from glycolysis in the cytoplasm will enter to produce enough energy through the TCA cycle and the electron transport chain. So this mitochondria is mighty because he's producing enough energy for our cell. So basically, these are the various major organelles in animal cells, and we want you to subscribe to our channel and keep following for more tutorials.